Hi, everyone. This is Mariam from Boston University. We are, now it's our final session of the day, oral presentations of selected abstracts. So all poster abstracts, all submitted abstracts are evaluated by our organizers and program committee and scored. And then these are 6 people who scored the highest. Our first talk is from Candida Barreto from Drexel University. And his talk is on neural efficiency of executive functions in children with and without Down syndrome.
Hello, everyone. First of all, I'd like to thank you for the opportunity of present this project here and for the great conference. So as Mary introduced me, I'm Candida Barreto. I'm a postdoc at Drexel University. And I'm going to present preliminary results of this very beautiful project that we have been developing at Drexel. So Down syndrome is a genetic condition that is characterized by the presence where a person is born with an extra chromosome. And I believe that everyone here knows somebody that lives with Down syndrome or some relative or friend or someone. And it has been reported among the major birth defects in the United States, and is among the most common single cause of intellectual disabilities. Also, individuals with Down syndrome present deficits in cognitive deficits when compared with mental matched peers. And although we have all these alarming numbers, understanding on these cognitive deficits are still limited. And in one particular domain that these individuals present this impairment is in executive functions. For those that are not familiar with this term, executive functions is a set of cognitive process involved in goal-oriented behavior. So attention, memory, inhibitory control. Like, for example, some people may be tired right now and want to just leave the room, but you're still here. Your inhibitory control is controlling you to just live here, right? It's also, executive control is related to the prefrontal cortex activity and in typical developing individuals. However, we don't know what happens with individuals with Down syndrome. We don't know how is this relationship between their brain functioning and these deficits in executive control. And this is related for the limitations of the available techniques, but as we discussed here today, neurotechniques has been advancing, and thanks for these advances. We now can use the functional near-infrared spectroscopy, which is a portable, non-invasive, and child-friendly modality to address this gap. Oops. So, so to do that, what we did was we used, we monitored the brain function of children with Down syndrome and without Down syndrome while they perform Arizona memory assessment for preschoolers and special population. It's very important to note here that this is a task that was developed by one of the PIs of this project, and it's special for this type of population. And in this task, we had like simple and complex conditions, and the simple condition, the participants were asked to tap on objects in the screen, and it's simple because all the objects were projected in just one screen without the need of alternation. In the complex condition, on the other hand, they had to choose in between 2 objects, and this required more cognitive effort, and also the stimuli were presented as alternate screens. Oops, sorry. So basically, we collected the performance of the task, and as I mentioned before, we use the fNIRS, and to analyze the fNIRS data, we did the normal pipeline of signal pre-processing by and then filtering the data and calculate oxyhemoglobin and extracted the average of the oxyhemoglobin for each condition and each episode. And then we applied the statistical models to compare the effects of the group and interaction effect. We also calculated what we call neuroefficiency index, which is a composite index where we combine performance and brain activity to see how efficient were the prefrontal cortex of those individuals. So our preliminary results, because this is a bigger project with more tasks involved, so here is just some part of the project. So from the behavioral perspective, we saw the results shown that for the simple condition, both groups, children with TD and with typical development and children with Down syndrome, performed very similar. However, for the complex condition, as you can see there, children with typical development performed much better than children with Down syndrome. And that's what we were already expecting, and it's nice that shows that our tasks were doing whatever we believed that it was supposed to do. And in terms of response time, we didn't find any group effect, but we did find differences in the level, showing again that our task was as expected. So higher response time for complex condition and lower response time for simple condition. So this is behavioral result, and then we move to the brain result, fNIRS results. So we found a group effect showing that children with Down syndrome presented a higher hemodynamic response than typical developing peers. And it shows like we may interpret that as more cognitive effort to perform the task. And we also found an interaction group, even though it didn't survive FDR corrections, but we could see that for the simple condition, again, and this is brain, this is not performance now. So for the simple condition, both groups had like similar brain prefrontal activation. However, when you go to the complex condition, you have a higher brain, higher prefrontal cortex response for the Down syndrome. And then when we go to the neuroefficiency, we found an interaction group between group and level, showing with, that in terms of efficiency, for the simple condition, both groups have similar performance. However, when you go to the complex condition, as we can see here, the group of children with Down syndrome present lower neuroefficiency than group with typical development. So in conclusion, we saw that children with Down syndrome presented a similar performance to TD children for simple but not for complex task conditions. And overall, Down syndrome group had significantly higher prefrontal cortex than the TD group. And also, we could find that lower neuroefficiency for Down syndrome than TD peers only during complex tasks. Overall, we could demonstrate that fNIRS may be a good tool to assess the brain activity of this population. And most importantly, as we are in a neuroscience of every day, this is one of the first studies that use this type technique for this type of population. And where we were discussing with the collaborators from the psychology department, and we were discussing about why is it important? why is it important to look into the brain activation? Is just say that it's activated or not. And she has a lot of experience in Down syndrome research, and she told us, no, that's important because so far we don't have any information like that for those population, and we need to find biomarkers for these cognitive deficits so that we can plan interventions to address these gaps. And again, that's thanks to all this development of neurotechnology that we are now addressing a very special population that were left behind for a while. And probably we're going to continue with more research on that and bringing new information for this population. I would like to thank you all for listening. I would like to thank you for the NIH and for our labs and for the families that participated in our study. Thank you.
Any questions to Candida? I have a question. What is your next step?
You mean in terms of the study?
Yes.
Okay, yeah. We have, so right now we have more data that we still need to analyze. We have some working memory data as well. So next step is compile all the information, turn it in publication, bring it to the community. But in long term, we also aim to continue with the studies in this population plan, like intervention studies, and see how this brain information can be helpful for this advancement in this field.
Okay, let's thank Candida and move on to our next speaker. So our next speaker is Nicole Eklund from Boston University, and her talk is on rhythmic auditory stimulation reduces prefrontal cortex activation during walking in people with Parkinson's disease.
Hi, everybody. Thank you, Miriam, for the introduction. Like she said, I'm Nicole Eklund. I'm a behavioral neuroscience PhD student here at Boston University, and I'm going to talk to you about how rhythmic auditory stimulation can reduce prefrontal cortex activation during walking in people with Parkinson's disease. So Parkinson's disease is the fastest growing neurological disorder worldwide. It's a very common neurodegenerative disease that affects all aspects of people's daily life. This survey asked about 900 people with Parkinson's, what aspect of their Parkinson's do they wish to see improvement in the most? This has all of the motor symptoms, as you can see here on the bottom, and walking is a top priority for people, and they wish to see improvement in that. So I know we had a great talk earlier today that talked about Parkinson's disease, so I'm just going to summarize a little bit kind of what it is. It's due to the abnormal aggregation of a protein called alpha-synuclein, which forms Lewy bodies within subcortical structures like the basal ganglia, the substantia nigra, as well as in the brainstem. Due to the formation of these Lewy bodies in the basal ganglia, there is a depletion of dopamine, leading to those typical Parkinson's symptoms that we know of, so the slowness of movement, that resting tremor, the postural instability. So right now, the gold standard of treatment is a pharmacological intervention to replace that depleted dopamine. However, Lewy bodies also form down here in the brainstem. As you can see, there are other neurotransmitters involved down here, and those dopaminergic medications are not as effective. The brainstem is responsible for those rhythmic, automatic movements like walking, something that we don't have to put actual conscious effort towards, so we're still searching for therapies to help that in people with Parkinson's. So on the left here, you can see a diagram showing that the healthy pathways, people have intact automatic control. So when you're walking down the street, you're able to take steps and not really think about those movements. You're able to do other things while you're walking down the street. You can text, you can talk to people, and your walking is not really affected because it's automatic to you. However, in people with Parkinson's disease, they lose that automaticity. So over here on this video, let's see if I can start it. Okay, you can see here, this is a patient with Parkinson's disease. He's walking slower, he has shorter steps, that decreased arm swing. We call that the loss of automaticity. I don't know how to stop the video, so I'm sorry, the sound. But that leads to gait variability, instability, and difficulty while he is dual tasking. So people with Parkinson's disease have to compensate for that lack of automaticity. So they bring in these attentional resources within the prefrontal cortex in order to actually carry out those previously automatic movements. So when the prefrontal cortex is activated, that means their attention it's difficult for them to shift attention to other things while they're walking, like dual tasking. So in order to help that, there's a very common rehabilitation technique called rhythmic auditory stimulation to help improve that automaticity. So this is the use of a rhythmic auditory cue that helps improve the rhythmicity of gait. So the internal cueing mechanism in people with Parkinson's disease is dysfunctional, but their external cueing mechanisms are intact. So they're able to lock onto that beat to bypass those damaged pathways. So it's activating more of an anticipatory process. They're able to expect that next beat with each step. I don't know if I can play this video. Yeah. Can I go back? Are you able to press play on both of these videos? So over here on the left, this is a person walking without the rhythmic auditory stimulation. So you can see your steps are fairly slow. There's a little bit of step variability there as well. And then if you could play the one on the right too, please. Thank you. So this is her walking to the beat of a song, and you can see that she's walking much faster, more efficiently, and you get to listen to a fun song while you're walking. And then if you could pause that next. Thank you so much. So we know that RIS can improve walking, but the impact on automaticity is unknown. So it's hard to promote a therapy or a technique when you don't understand the underlying mechanisms behind it. If RIS is an attentionally demanding task, we don't want to encourage people to use it because that's not really helping make their walking more automatic and more sustainable. We want it to be more efficient. So for our study, our first aim here was to examine the effects of RIS on prefrontal brain activation during walking in people with Parkinson's. We hypothesized that compared to single and dual task walking, RIS will reduce that prefrontal cortex activation and reduce that attention. We're interested in looking at the comparison between dual task walking and the RIS because dual task is a known attentionally demanding task. So we want to compare RIS to see if this is an attentionally demanding task or if it's the same level of cognitive load as a dual task. We're also looking at the effects of RIS on spatiotemporal gait parameters as well to make sure we are improving their walking in addition to the reduction of attention. So compared to single task walking, we hypothesize that it will improve their cadence, stride length, and speed. So we brought in 17 people with Parkinson's disease for a single study session, and we measured their changes in oxygenated hemoglobin using functional near-infrared spectroscopy, or fNIRS, during walking conditions. So this was our walking paradigm. We had people walk for a little less than 10 minutes 3 times, and it alternated between single task walking, which is just their usual walk, and we didn't give them any additional instructions, and then one of the experimental tasks. So we looked at dual task walking, which we had them subtract 3 from a random 3-digit number, RIS at 100% of their cadence, so essentially their same walking speed, and then RIS at 110% of their cadence. So slightly faster. Studies have shown that an increased tempo actually shows more improvements in their walking, so we wanted to look at that as well. Next. So this was our fNIRS setup, as we all have talked about today, but it's a portable, non-invasive brain imaging technique that makes it great for walking tasks. They're able to carry the device with them as they're doing the task. We measure changes in oxygenated hemoglobin from the previous task. So for us, our reference task was the single task walking. We had 8 regions of interest, so all of these frontal regions in here that are colored, we have inferior, middle, and superior regions on both the right and left hemispheres, and then we had 2 premotor regions, but we're only focusing on the colored regions just for this analysis. So we found differences in prefrontal cortex activation between the dual task walking and the RIS conditions. So over here I have one of the ROIs that we looked at. In the blue, you can see that the dual task walking increased activation as we expected. And then you see in yellow that the RIS at 100% decreased the activation, and then with RIS 110 decreasing their activation even more. as you can see, the rest of the ROIs also followed the same pattern. So with dual task walking increasing the activation, showing that it's more attentionally demanding, and then with RAS decreasing, with 110 with the greatest decrease, aligning with our hypothesis. We also found changes in spatiotemporal gait parameters compared to the single task walking. So over here on the left, we have the cadence. So with dual task walking, it showed a decrease in cadence. And then with the RS 100, we didn't see much of an increase because it was similar to their usual walking. And then with RS 110, we saw a greater increase. This makes sense because we manipulated this and we did force them to match that faster cadence. In the middle over here, you can see that their stride length decreased when they were subtracting. So they had shorter strides, less efficient walking. And then we had increase in stride length for both RAS conditions. So we lengthened their strides during the RAS. And then combining these, we got an overall increase in speed during the RAS conditions, and then a decrease in speed during the dual task walking condition. So in summary, RAS walking reduced PFC activation compared to single and dual task walking, suggesting lower attentional demands. The gait metrics also aligned with the PFC activation pattern, suggesting that the RAS provided a more rhythmic and automatic walking. And faster RAS tempos showed trends toward greater benefit. So the 110% showed greater benefit to their walking, highlighting the need to explore the tempo-dependent effects. And so some clinical implications. So RAS may enhance gait automaticity in people with Parkinson's disease by reducing their cognitive load, so reducing that attentional demand. And improvements in gait automaticity will hopefully promote safer and more sustainable walking in the real world. We want people to get out there and walk and exercise and feel safe while they're doing it. And then studies with longer walking periods are needed to understand more of the real world implications. So just a thank you to our great team, as well as our funder, the American Parkinson's Disease Association, and then the BU Neurophotonics Center for their support. So, thank you.
Any questions to Nicole?
I have a question. What's the effect on turning?
So that's a good question. We haven't fully looked into that yet. We have the equal amount of turns when we have them do their walking tasks, so we kind of assume that it just cancels out when we average everything out. But yeah, that is something we need to kind of parse out eventually.
Any other questions? Let's thank Nicole again and move on to our next speaker. Our next speaker is Morgan Fogarty from Washington University in St. Louis. And she's going to talk about leveraging fMRI datasets to simulate and evaluate the potential performance of human optical neuroimaging systems.
All right, hello. Thank you so much to the organizers for this opportunity to present my research and also just for the wonderful conference so far. I'm Morgan. I'm a postdoc researcher in the Neurophoto Lab over at Washington University in St. Louis. So the gold standard for functional neuroimaging is often considered to be fMRI, which provides these high-resolution images of the brain. fMRI researchers have highlighted the importance of imaging the brain during rest or even everyday tasks, for instance with movie watching or listening to podcasts, as ways to study the brain during natural settings. However, fMRI is collected in a limited research setting where the participant must lie down and remain still throughout the duration of the scan session. Instead, we need to move towards more naturalistic imaging devices to do studies outside of a traditional scanner. And so from our lab, we propose the usage of diffuse optical tomography as a viable functional neuroimaging technique that can allow for in-person interactions or even become fully portable for imaging in increasingly naturalistic settings. So briefly, this optical imaging technique works by shining light at varying wavelengths onto the scalp, and this light travels in this banana shape that we can see here between the source and the detector, and therefore through our tissue. The light absorption of blood can then be used to map functional brain activity similarly to that of fMRI. Using this method, we can see 1 to 2 centimeters into the head, resulting in visualizing cortical activity. the arrangement of these sources and detectors also has a direct relationship on our image quality as well as our field of view. These high-density DOT systems are the primary focus of our research lab, which involves packing many optodes onto the head to get image quality similar to that of fMRI. And so what we say is that we can use HD-DOT as an alternative to fMRI for these naturalistic settings. But with optical neuroimaging, we have many important decisions that we have to make. So for instance, we have to decide where we're going to be placing these sources and detectors onto the head in order to cover our desired brain regions. Additionally, we need to decide stimuli that are both engaging, but also more importantly, elicit activations that are within our field of view. As we start to build out larger data sets, we can also think of things such as how much data is going to be required to achieve desired accuracies for our models of either age or cognitive prediction. So these early experimental decisions are really crucial to developing a successful new imaging system or study design. And while we can use literature reviews to answer many of our questions, a method to quickly and easily kind of simulate these results would be more efficient. So while there's work being done to develop these large-scale data sets using optical imaging techniques, these data sets tend to focus on either specific populations, and FNIRS and DOT does often lack standardization in equipment and data processing across research centers. So therefore, we turn to these large-scale fMRI data sets, such as those uploaded to N.E.W, which is a platform for sharing neuroimaging data that requires data be uploaded in a standard format. And this standardization is what really helps the data be accessible. So as we can see here on this plot, there's an ever-growing number of publicly available data sets available on N.E.W, many of which are fMRI data. This plot ends in 2023, but as of earlier this year, we had about 1,200 data sets on N.E.W with about 55,000 participants. These fMRI data sets also range quite a bit in terms of amounts of data, amounts of participants. And so what we can see here is that we can look at hours of fMRI data per subject on our x-axis and then number of subjects on our y-axis. And this diversity in data sets is really useful to us. So for instance, we can look at studies such as the UK Biobank, which has about 10,000 participants, but with fairly minimal amounts of fMRI data. But this can be particularly useful for looking at these age or cognitive prediction models and determining which arrays are going to be capturing data within the field of view that we're interested in. Similarly, we can look at studies such as the Midnight Scan Club, which has many hours of data collected in the same participants. And this can be useful for looking at more niche tasks such as decoding or other naturalistic studies. So since the endogenous contrast for the fMRI bold signal is also relative changes in hemoglobin, we can use existing data processing tools to simulate optical measurement data from the fMRI bold signal. So we can think of this problem as simply as y = ax. So we have our fMRI data, our x, and we're trying to solve for y, which is going to be our simulated optical measurements. To do this, we just need to construct our A matrix, or our sensitivity matrix, which relates relative changes in optical measurements at the surface to relative changes in absorption in the volume. So in this context, we can think of this A matrix really as a way to map between voxels and optical measurements. So generating the A matrix is really the most crucial step in our simulation pipeline, and it's where we're going to define our virtual imaging system. So following the neuro.toolbox, we can start by generating a mesh using a 5-layer segmentation from T1 and, if available, T2 MRI data. The desired optode grid can then be placed onto the mesh for the near-fast computation of the Green's functions to form our A matrix. Once we have this A matrix, we can compute the simulated measurements through y = ax, and these measurements can then be reconstructed by first inverting and smoothing the A matrix, and then multiplying the A matrix by our simulated measurements in order to get our simulated optical data. We can then do further post-processing to answer the experimental questions that we're curious about. So for the simulation work, we'll focus on 3 virtual imaging systems. So we have our very high-density DOT system, high-density DOT system, and a whole-head sparse fNIRS system. The VHD DOT system contains nearly 10,000 measurements from 255 sources and 252 detectors, whereas our HD DOT system has around 2,500 measurements from 128 sources and 125 detectors. And then our whole head FNIR system has 32 sources and 32 detectors, resulting in around 100 measurements. And so what we can see here as well is we can do comparisons, not just of the number of measurements, but also the percentage of the cortical areas that we can see with our imaging systems. And then also we can look at our image resolution by computing the full width half maximum of our simulated point spread functions. And we can see how that varies across our 3 different imaging systems. But while image quality and field of view are obviously quite important, there are trade-offs to consider about wearability and resolution of our imaging systems. So while the VHD-DOT system provides high-quality images across much of the superficial cortex, the system itself is susceptible to noise from motion and is clearly not portable, as you can see from the picture there. Alternatively, FNIR systems, while sparse, can allow for imaging in increasingly naturalistic settings and are lightweight enough to be used for populations such as children. When considering an experimental design, it's essential to consider all aspects from data collection to analysis to ensure the imaging system is well calibrated to the intended tasks. So using subject-matched VHD-DOT and fMRI data, we can validate our simulation methods by comparing the simulated DOT measurements from fMRI to collected DOT measurements. So these group average maps have substantial overlap between the simulated and the collected DOT data when thresholded at 50% of the maximum value. Here we can quantify further the overlap using Dice coefficient and also compute the spatial similarity of the collected and simulated maps using Pearson correlation. This overall really validates our simulation methods and showing that they are capable of capturing the field of view and the image resolution of optical modalities. So what I'm going to switch to next is looking at a case study using these simulation approaches. So in our case study, we're interested in semantic brain mapping using diffuse optical tomography. Foundational research from Dr. Alex Huth has led to the mapping of semantic concepts across the cortex, as we can see in this video. In this experiment, participants listened to 10 to 15-minute narrative stories from the Moth radio hour while lying in an MRI scanner. Using word embeddings to extract semantic meaning, a regression model was then used for voxel-wise encoding. More recent developments of this work have extended the dataset to include nearly 16 hours of listening data in single participants. This has led to the resynthesis of speech from fMRI data from this foundation of semantic mapping. With DOT as a wearable surrogate for fMRI, we're interested in extending these techniques to a wearable imaging device that can be used in more naturalistic settings. But before we go off and collect 16 hours of data in single participants with our imaging system, we want to first evaluate if this is even feasible given the field of view of DOT. So we took to OpenNeuro because these data sets were publicly available, and we were able to simulate the optical measurements for the task of semantic encoding. So in blue, you'll see highlighted where like which parts of this plot are from OpenNeuro. And so these MRI data were then used to simulate optical data for our 3 virtual imaging systems, so that fNIRS, the HD-DOT, and the VHD system for 27 Moth radio hour stories. These simulated measurements were then used to fit an encoding model, with 26 of the stories being using code from GitHub, which is indicated in green. The encoding models were then validated using a held-out story where the measurement time traces were predicted using the encoding model generated time traces and compared to the simulated DOT time traces. And so what we can see here is that the correlation is highest in frontal and auditory regions associated with word processing, establishing that semantic regions can be captured by this encoding model. So we can see it in MRI up in the corner there, and then for our 3 virtual imaging systems. These regions include the temporal cortex, parietal cortex, and prefrontal cortex, which display a higher prediction performance than the other regions, such as occipital cortex. The virtual VHD and HD-DOT systems offer similar areas of high prediction performance, indicating that these systems can capture semantic regions. In addition, we compared the maximum correlation values for each of these systems, and again, we see the VHD and HD-DOT performing similarly. We can also evaluate this as the number of voxels that have correlations greater than 0.2, and also the percentage of cortical voxels that have correlation greater than 0.2. And again, we can see that HD-DOT and VHD-DOT are performing fairly similarly, with fNIRS not having sufficient coverage for this type of task. So in conclusion, the simulation approach is beneficial for selecting ideal optode configurations. Prior to system development, it can be used to evaluate new stimulus protocols, estimate our necessary data collection, and evaluate processing pipelines on simulated results while collecting our optical data. This connects to studying the brain in naturalistic environments as we establish optical techniques as a surrogate for fMRI and begin to extend optical methods to studies outside of a traditional laboratory setting, such as our example here with piano playing and conversation studies. So with that, I'd like to thank all the co-authors on this project, in addition to our funding sources. I also want to shout out 2 PhD candidates from Joe's lab that are also here presenting their research. Vita Fenner presented her work on decoding of naturalistic movie clips with a semantic model using HDDOT at the poster session earlier today. So if you're interested in the semantic stuff I talked about, I highly recommend talking to her at the networking event later. And then additionally, Will Hammock will be presenting his work on the wearable HDDOT system during tomorrow's poster session, and I also really recommend checking that out. So with that, any questions?
No questions? Let's thank our speaker again. So our next speaker is Jason Nan from University of California, San Diego. And his talk, is on a novel neurofeedback task targeting depression and rumination symptoms.
Hi, everyone. So my name is Jason. I'm a postdoctoral scholar at UCSD under N.E.W. Labs. And first, I'd like to thank the organizers of BU for having me talk and awarding me a travel grant. So just a little bit of background. Depression and the kind of state of mental health can't be understated. We need much more research into it and also develop better treatments. For depression specifically, the 2 most common treatments given are prescription drugs or transcranial magnetic stimulation. And we've heard earlier today about how these are kind of not very optimal solutions anyways. But just going over it briefly, they're both very expensive. Prescription drugs have a lot of nasty side effects, like actually increased anxiety, cases of suicidal ideation, and just making depression worse. A lot of that is due to the fact that from a neural cognitive aspect, we don't really understand how these SSRIs are treating depression. We only know from a pharmacological aspect. So it could have a lot of disastrous side effects. Transcranial magnetic stimulation, or TMS, we do have a target for it, the DLPFCA, but that's also very expensive and has the additional burden of it being in clinic use only. So the goal of my project is to develop a more accessible and neurophysiologically targeted intervention for depression. There will be 3 parts to this talk and kind of for this project overall. Part 1 will be finding a neuromarker that is linked to depression. The second part will be designing a system to train individuals to modulate this signal. So kind of answering the question of, well, once we have the target, what are we going to do with it? And the third part will answer the big questions. Are people actually able to modulate this signal? And is this even useful? So moving on to part 1, finding this non-invasive neuromarker. Okay. So we'll base this search from this on a breath attention task that our lab had developed. It's called 2-tap, and it's very simple. Just close your eyes, and every 2 breaths, tap the space bar. In the first initial paper, we had found that neural activity during this task linked very strongly to traits of mindfulness in source localized space. So we wanted to develop kind of an EEG way to operationalize this marker. And so for this, we look at alpha band imaginary coherence. Now, since this is an eyes closed task for rumination and like meditation kind of, the primary signal will be alpha band activity, and that's why we're doing that. We're looking at imaginary coherence instead of just raw connectivity because this minimizes the effect of volume conduction, which can happen when you record EEG. So in our search, we wanted these imaginary coherence values to relate to all mindfulness, depression, and anxiety. Furthermore, the effect of mindfulness should be opposite to that of depression and anxiety, because increased mindfulness is good and decreased depression and anxiety is good. So in our search, we looked for all possible combinations of 2 electrodes using our 24-channel setup. And we found that FCZ and FC4 alpha band imaginary coherence remained significant when we FDR corrected for all possible combinations, as well as the link to all 3 traits. And so just looking at here, we validated increase of FCZ, FC4 imaginary coherence leads to increase in mindfulness, while the reverse is true for depression and anxiety. So now we have a very robust signal that comes from a purely data-driven way on how depression, anxiety, and mindfulness can present in the brain. Now moving on to part 2, how do we design a system to train individuals to modulate this? And from the title, I assume you guys can guess that it's neurofeedback. But the requirements for about why we wanted to use neurofeedbacks is actually that it's very non-invasive, so just recording EEG, and it doesn't require any pre-approval, and it's very simple to set up. We also wanted it to be very accessible, both in terms of cost and this idea of self-sustainability, and that's the idea that anyone can just go in and elect to do this without having to jump through a bunch of hoops. We also wanted a system that is very low risk, which ties into the accessibility. And then this other idea about it developing portable skills. So if you can train an individual to modulate their activity, volitionally, they can take it anywhere into their daily lives. If they're walking on the street, catch themselves in a ruminative cycle, they can just practice what they learned during the neurofeedback sessions. So just going over neurofeedback very generally, what we're doing is we're recording brain activity with an EEG. We're sending that signal to the computer. The computer is parsing out this FCZ, FC4 alpha imaginary coherence, and then it's modulating a sound from the computer. The sound is then heard by the participants, and then they can adapt their strategy to try to increase the volume of the sound. Now, how is this different from the traditional neurofeedback paradigm is that since this signal was developed from a behavioral mindfulness task, we wanted to include this into the system and keep it in a trial-based manner. So we'll specifically instruct the individuals to focus on their breath in order to try to increase this signal. We'll also be doing it in a cognitive task approach where we'll give them multiple trials with 2 forms of feedback that I'll go into in a little bit. And then these 2 forms of feedback and just the attention to breath, all of it is supposed to synergize together and help them achieve performance. So going over the general task structure, this looks very similar to any neurofeedback and cognitive task design. We'll first do all of our initializations, calculating a baseline, and then we'll go into these 5 trial blocks. So each block will be 5 trials that they'll complete back to back. Each trial will last 20 seconds. And then in each of the 20-second trial, the volume of the meditation music they're hearing will be updated every 200 milliseconds. Now, this volume scale is discrete increments, and so having it at a fast pace of 200 milliseconds makes it seem more continuous. At the end of the 20-second trial, they'll be provided with a secondary form of feedback, and this will be based on their performance during the entirety of the 20-second trial. And you can see the different rating scales that we have there. So from our previous research, we actually found that having these 2 different timescale feedbacks actually promoted learning. Having this really fast feedback, the 200 milliseconds, can help individuals fine-tune their strategy. The overall feedback at the end of the 20-second trial can help them tune just the overall strategy of what they're trying to achieve. And if this sounds very complicated and just what the hell are they doing, it's very the neurofeedback is very personalized, so there's no one right action that they can do. We tell them to focus on their breath, but some individuals have reported that they focused on the inhale, and then other participants have said that they have focused on the exhale. So it's about finding out what works for each person. Going over the study design, we'll have 3 arms in this. We'll have active neurofeedback, sham neurofeedback, and also the no contact control. All 3 of them will get pre and post surveys 4 weeks apart, while the active and sham neurofeedback group will be assigned 5 of these in-person neurofeedback sessions. Each session will be preceded by 2 days of at-home breath training, and this is to make the subject recruitment and task a little less burdensome on the participants. So instead of having them come in for 15 days, we'll only have them come in for 5 days, and they'll just do at-home breath work 2 days prior. The sham neurofeedback group will be provided with a sham signal, so they'll be provided with an imaginary coherent signal that they're actually modulating, but it is not FCZ, FC4. The at-home breath training will also be a sham version of the breath attention task. So in terms of subject recruitment, right now for the active group, we have 19 enrolled and 18 of them completed. We're aiming for 20 in each arm. The no contact control, we're just started recruiting, and we have 7 enrolled, but none of them completed so far. And then we still need to start recruiting for the sham neurofeedback. So now that we have the first 2 parts, let's go on to the big questions. So does this even work? And the answer that we think is yes. So what I'm showing here is the every single trial that individuals have performed and their FCZ, FC4 imaginary coherence over the entire trial for across the 5 days. So each day is in a different color. And I'm showing the mean across all of the subjects as well as the standard error. So as you can see from each of the days, there's an initial drastic increase, and then it kind of plateaus out for the remaining of the trials. The interpretation I have right now is that people need a couple of trials to get in the zone, but once they're able to get in the zone, they're able to sustain that pretty well. Then we also see that imaginary coherence increase across days. And then this is further confirmed by a linear mixed model, where we took the imaginary coherence and tried to relate it to the main effects of trial number and the number of days, and they're both significant. Okay, now we can look at the symptoms of depression, anxiety, mindfulness, and rumination to see if there's any translational benefits. And again, the answer is yes. So if we look at depression, anxiety, and rumination, we see that all 3 has significantly decreased at the end of the study, while mindfulness has significantly increased as well. Furthermore, if we take a Spearman's correlation between the reduction of depression and then the value of the raw coherence across all of their trials, we see a significant negative relationship. And what that means is the more they are able to raise this raw coherence or imaginary coherence value, the more reduction in depression that we see. So ability to perform the task is linked to a more drastic reduction in depression. Now just going over the summary of what I went through. First, we identified that alpha band imaginary coherence was linked to depression, anxiety, and mindfulness in a very robust and data-driven manner. We developed this novel neurofeedback task paradigm to train individuals to upregulate this signal. And we showed that the individuals can actually volitionarily increase this signal. And we saw improvements in depression, anxiety, rumination, and mindfulness. And furthermore, that the ability to modulate this FC4, FCZ imaginary coherence is linked to improvements in depression. Now, just going into the future directions, this is a open label pilot study to see if this paradigm is even worth pursuing. And the next step would be to, for this one first, we need to find the sham signal to recruit the sham group then. And then also a randomized control trial will be next. This initial pilot study showed that this is worth pursuing. And the last point is I want to investigate if individuals can modulate this signal with no feedback. And then this ties into the idea of neuroscience for the everyday world and the implications. So once individuals have learned a strategy, then hopefully they can learn to implement it in their daily lives without any feedback or having to go into clinic. So they catch themselves in a ruminative cycle, start practicing the strategy that they learned in the in-person neurofeedback, and hopefully it will help break them out of the cycle. The next implication that I want to propose is that it's not just limited to depression and rumination and anxiety. As long as we have a good, robust neural signal, then we can adopt this paradigm to kind of train individuals to upregulate any signal related to any symptom morphology. And with that, I'd like to acknowledge my mentors, Jyoti Mishra and Dr. Dakshan Ramanathan, and the team at N.E.W, as well as everyone who helped with this project. Also like to shout out the organizers of this committee again for having me speak. And then I'll open it for any questions.
Question. I think I missed this when you were explaining, but when you identified the imaginary coherence band, did you search everywhere in the brain and that's where you ended up? Because that makes it really hard for you. I'm trying to figure out what the sham would look like and what the application would look like if it's a very specific part of the brain in terms of coherence. Like, help me understand that translation piece, because I feel like I missed that.
Okay.
Where did you look and what did you find and why does that matter?
Okay, great question. So for the imaginary coherence, so we're recording this EEG from 24 channels, like using a modified 1020 version of the of an EEG cap, and then we looked for every single 2 combination electrode pairs. So 24 choose 2 combinations of electrodes, and then we wanted whatever signal that we found to relate to mindfulness, depression, anxiety. And so when we searched around all 24 choose 2 combinations, we get a lot of like FDR corrections. So then we FDR corrected across all of these possible combination pairs. And then the electrode that remained significant or the electrode pair that remained significant was FCZ, FC4. So that was our target signal because it was then linked to like depression, mindfulness, and anxiety. So higher values of this imaginary FC4, FCZ, I'll just call it target signal, was related to lower depression and lower anxiety and higher mindfulness. So then we wanted to train individuals to modify or modulate this signal towards that higher end to hopefully reduce their symptoms of anxiety, depression, and increase mindfulness. Now, for the sham neurofeedback, what we want is the complete opposite of this target signal. So we want something that is completely like neurally neutral. So it's not related to depression, anxiety, or mindfulness. Then the sham neurofeedback, we would have individuals learn to modulate that signal. And then the goal of the sham neurofeedback is to say, okay, well, if people are doing this mindfulness task, they might just improve in depression, anxiety. We wanted to link to the specific modulation of FC4, FCZ that is linked to this improvement in depression, while just if they're modulating a random neurally neutral signal, then we ideally won't see improvements in these metrics.
So the modulation technique is not different?
Yeah, the modulation wouldn't be different. They would still be given the feedback, except that the feedback signal would be tied to this, this sham signal.
I have a question.
Yes. Oh, there's also a question in the back.
Yeah, so did you think of having another control which does the breath exercise without neurofeedback at all?
Yes, so that would ideally be in the randomized control. We do hope to include another arm where they just do at-home breath training, but not this neurofeedback. Cool. But we do have a previous paper that had actually implemented this. It was a variation of this breath attention task called mindfulness training. And they found that just doing mindfulness training alone wasn't linked to a reduction in depression or anxiety. Mm-hmm. So we believe that with the addition of neurofeedback, that we are seeing an effect now.
Okay, cool.
Okay, isn't depression and rumination often associated with excess coherence in the brain due to just reduce differentiation between different in the brain?
So yes, the idea of coherence and depression, I mean, it's very muddy. There's not a ground truth of what is happening. We are specifically looking at alpha band imaginary coherence during this meditation task. And then the theory behind it is we are recording from this like EEG setup and we're doing our specific re-referencing scheme. And just all of these things can actually affect what neural signature that you are seeing. And the theory is that we're just recruiting a large cohort. We have 200 something subjects in there and saying, okay, well, in our specific use case in this task, in this recording setup, we have this FCZ, FC4 imaginary coherence, and that is the signal, or that is our measured signal that relates to depression and anxiety. And then we wanted to create a paradigm that made it actionable.
Okay, thanks, Jason. Our next speaker is Martin Zeber, University of California, Los Angeles, and he's going to talk about scalp EEG signatures of human hippocampal theta during real and imagined navigation.
Thank you. Yeah, my name is Martin. I did this work at UCLA, but recently I joined Duke University, so it's not a typo or a mistake. I'm very excited to be here, not only because of the great scientific talks of today, but also because of the great view and great company this conference is taking place in. So today we talk about scalp EEG signatures of deep brain signals from the hippocampus during real-world imagined navigation. I will start my talk. I will start my talk with. I'm not sure if I turned it off. Maybe I did. Okay, there we go. With my excitement about navigation and imagination, and I'm interested in the parallels of these tasks, because if we understand the brain processes of these cognitive tasks better, this does not only tell us more about how we find our ways in the real world, but also potentially might inform us to better understand the neural processes underlying experiencing or recollecting already experienced episodic events in our past life, but also to envision the future. And we already know very well about the brain structures involved in these tasks. That is the medial temporal lobe or the hippocampus. So we know about the functional anatomy, but also we do know about the computational units underlying these processes, such as spatially tuned cells. What we know less is how we combine these functional units to produce meaningful actions to navigate the real world. So one particularly interesting element or neural mechanism that binds these computational units together are theta oscillations that provide temporal structures to neuronal firings. So theta oscillations are proposed to provide temporal structure that then results in these well-organized neural firing patterns. And these neural firing patterns are not only observed during running in animals, but also in periods where animals just run in a wheel between their navigational tasks. So then the suggestion was that animals then presumably think about their future trajectories. But that is, of course, hard to prove. But what we actually can do with humans, we can ask them what their experience was during imaginations, or they just simply report their experience during imaginations to us. So what we did in lab is designing an experiment where people learned to navigate particularly defined spatial routes in our lab space. Oh, sorry. And after learning these routes, they were coming back to the treadmill and only imagined walking on these routes. And we also had access to their neural activities, because we could record directly from the hippocampus using the RNS NeuroPace device. So we had chronically implanted intracranial EEG electrodes, which then provides us with intracranial EEG recordings during these real-world tasks. And simultaneously to these intracranial recordings, we also have scalp EEG. So now we can ask the question, what can we observe in which modality, and potentially combine both techniques in order to study the whole brain while people did these very realistic tasks. So what we first looked at is that their neural activities depending on these spatial routes. I'm showing the top-down view of the spatial routes they learned on the top left, and the neural activities on the right. And you can see here in the time frequency activities already with naked eye that in a specific frequency range, in the theta range, these oscillations go up whenever participants are approaching turns, which are indicated in these vertical lines. So then we looked at these theta oscillations, and we can then combine the neural activity and the spatial trajectory, which I'm showing here on the panel below. And you already can see that when we combine motion tracking and neural data, we very easily can observe that these theta oscillations are depending on their position in these segments. So the roots are composed out of 5 segments, and whenever a participant is approaching a turn, theta oscillations go up. And that also we observed during imagined navigation. So here during imagined navigation, they were walking on a treadmill, and they were not actually walking through space, but they were just imagining themselves walking on the same routes. And we saw very similarly temporally structured modulations of theta oscillations during the imagined navigation. And how we did this plot is just by warping their neural activities from the start to the end of their imaginations here. And also furthermore, we quantified this relationship more specifically, where we have like highly structured temporally structured theta oscillations during the walking condition. Nothing of significance during the control condition is just walking on a treadmill without any additional task, and with something in between during imagined navigation. So we see temporal structure of theta oscillations only while they imagine their navigational experience. And this also we can visualize. Please help me starting this video if you have a second. Oh, like if you would start this video for me, please. So what we show here is the behavior on the top left, the raw EEG signals on the bottom, and theta power on the top right. And you can see whenever a participant is approaching a turn, signals get stronger, more oscillatory. And this is what you can see already in the intracranial raw data. You can see like the last turn. An important observation was even before people were approaching these turns, we already saw theta oscillations going up before we had any behavioral signature of their turning behavior. So this data was quite exciting to us. So we wanted to go further and ask, can we not only study the hippocampus, but potentially the whole brain while we are moving and experiencing the real world? And that is this work is based on our previous work where we combined scalp EEG and intracranial EEG and asked the question, can we actually reconstruct intracranial signals from the scalp? So we had high-density scalp EEG recordings. We knew exactly where the electrodes are. We have a model describing the electrical relations between the brain and the scalp. And then we can invert this relationship to compute a map and then compare which point of this estimate is mostly correlated with the ground truth that we have recorded in the intracranial recordings. And we showed that this is possible for the thalamus, which was very exciting to us, but others were showing similar things or similar results for the hippocampus. So we asked ourselves, so if we have all this previous knowledge, can we then combine both technologies? So first of all, it's important to say it's very important to clean artifacts or correct for motion artifacts while people are moving in the real world in the scalp EEG. On the top, I'm showing raw scalp EEG data. On the bottom, I showed corrected data. And the way how we corrected the data is we informed the scalp EEG recordings with the intracranial signals. So we only look at signals that we recorded in the scalp EEG that are correlated with what we measured in the intracranial signal. And when we look at topographies, this becomes even more clear. On the top right, you would see that there are very noisy topographies in the raw data, but on the bottom right, this is then the corrected data, but it's a restriction to only functionally related signals that are related to the medial temporal lobe areas. And then we can look at the differences whenever there is a theta bout or not. So we just simply measure whether there's a theta bout in the intracranial signals and look whether the scalp potentials get stronger. And that is actually the case, is what we show here. So that works out for theta bouts, shown here on the top left, but also for increased theta power. So whenever there is like low theta power, we see a lower scalp potential. Whenever there is high theta power in the intracranial signal, we see higher scalp EEG potentials, which already indicates that we do see these intracranial signals or these deep signals coming from the hippocampus on the scalp. And the topographies are also specific. If you do this comparison for a left electrode, we see left lateralized topographies, and if you do it for the right, it's different. And then we can do the source analysis, as I introduced before, and we see whenever we compare to the left electrode, it maps to the left. So here I'm showing, this is the ground truth in blue. This is the intracranial electrode that was implanted, or the location. And what I'm showing here in yellow is where the non-invasive map would point to, meaning if those are overlapping, we found the correct signal or the correct location. And then to quantify this relationship a little bit further, we computed the lateralization index to quantify if left signals are mapping to the left MTL and right signals are mapping to the right MTL. And that is actually the case, which I'm showing here on the top right panel for 17 out of 18 channels we had in our study. And what I'm showing here on the lower right is the correlation between this global field power effect and the lateralization, meaning whenever we see strong scalp potentials, we are also able to correctly localize the effect. Whenever we do not have a strong scalp effect from an induced theta bout, we're not able to reconstruct them. So meaning if you have a very strong signal in intracranial signals that spreads to the cortex, we also can reconstruct it. But if we don't have any kind of like physiological meaning signal, then it's impossible to get it or reconstruct it. And here I'm just showing a little bit more individual data. So different people have different implantation sites, but what I wanted to highlight here is first more topographies for each individual, but also to demonstrate that depending on the implantation site, also these functional reconstructions, they seem to follow the ground truth. And now we can ask more interesting questions, going back to the experimental question we introduced before, looking at a conditional, like experimentally conditioned differences. So here I'm showing the comparison between intracranial and scalp EEG results when comparing imagined and real-world navigation. And what we showed in both modalities is that relatively in the left and right medial temporal lobe, that the right medial temporal lobe activities seem to increase during imagined navigation in comparison to real-world navigation. And you also can see here that corresponds very well to the source localization of the non-invasive data. So this is the invasive, and this is the non-invasive, and this is the non-invasive sources. And also what we then can ask is, can we see any activities outside of the medial temporal lobe that is different between different conditions? And what we observed is when we compare real-world navigation versus imagination, is that medial temporal lobe activities increase. And that was exciting to us because now we have an observation that we know these signals are significantly correlated to medial temporal lobe activities, but there we never had an electrode. So somehow through the combination of intracranial and scalp EEG, we can investigate brain areas where we actually never had an electrode in. So for a follow-up experiment, we can then test the meaning of this prefrontal cortex activities that are presumably related to the generation of actions, because we act upon our spatial maps during real-world navigation, which we do not do during imagination. So that is a working hypothesis we are working on right now. So with that, I would like to summarize that theta oscillations are temporally structured during real-world and imagined navigation. They're internally generated because during imagination there are no environmental constraints we can use. So they're internal generated theta dynamics. They might support imagination and episodic memory, recollection at minimal sensory input. Scalp EEG contains activities of deep sources that can be reconstructed using source imaging techniques. And the combination of intracranial and scalp EEG might enable us to work towards novel perspectives to study cortical but also deep brain activities in mobile setups. And having that said, is we can use both modalities to get to this subspace I was introducing before, and using these analyses, then we might be able to enlarge our population size of included participants of our study. So we might be possible to not only study, in this case, 5 participants with intracranial electrodes, but more participants only with scalp EEG, or by combining both is the most interesting one to me, because then we might be able to study functionally related areas that are informed by the intracranial signals. So for instance, one application could be we could study the functional effects of deep brain stimulation on the cortex. And with that, I would like to thank my lab, my great supervisor, Nanti Susana. Matthias was part of the study, and all other lab members. I would like to thank both universities, UCLA and Duke, as well as our funding agencies. Please contact me or reach out. I'm happy to answer any question. Thank you.
Really cool talk. Thank you very much, Martin. I know you didn't present any data on connectivity, but can you speculate whether this type of data would then let you look at the connectivity to see where the hippocampal theta is progressing to other areas?
Yes, no, I mean, this is what we're heavily working on. So far, it varies, like the connectivity analysis, depending if you focus on phase or amplitude modulations, they vary a lot across participants. So we're not yet sure, but we, of course, like follow up on that. What I can say is that usually analysis, looking at, where the bouts are before or after, like looking at amplitude modulations, seem to be yielding the more robust effects. And very presumably, it's rather hippocampus to PFC than the other way around.
Can you use the low frequency information from invasive electrodes as and treat it as if it were a scalp electrode to look at better localization for scalp EEG?
Yeah, that's somehow like one of the aims of the study where we combine, but also there's an idea to inform the localization procedure. Yeah, that's one of the directions we do want to go, yes. But so far, I mean, the models are like decent. And so the idea is to have like these few points that inform then the analysis, and the other points in the brain, like the PFC, we can't be absolutely sure. But the line of argumentation is if it works for these very difficult regions like the MTL, probably the cortical areas are correct too, because that's like an easier problem to solve. If that makes sense. Okay. Thank you. Great question.
Thank you very much for the talk and the elegant study. I was a bit curious. In the last slide, you wrote that the theta dynamics related to those turns were temporal. They had temporal patterns, but I was wondering if they were also linked with the space, so how far from the turns the participants were.
Yeah, no, yeah, of course, if you would just look at the real-world behavior, they would be spatially modulated. But I'd say they're like temporally structured, because that is a more general statement, just thinking about the imagination data we found. So this temporally structured quality of theta oscillations is the most important finding to us, because theta oscillations are not continuous in humans. And the crucial finding was that the specific moments of these amplitude increases are the most informative factor, not only during spatial navigation, but imagination too. So there must be something that is like more general, that is not only space. It might be space, like imagined space, right? But like, yeah, this is why I was like saying it's not only space. Like, it's not necessarily depending on whether you're close to the turn or not. You can be close in your mind, but not physically, right?
I was just wondering, in the slide you're showing imagination against real walking, it looked like it was sort of maybe spreading into the retrosplenial cortex. Does it extend like more superior?
Yes, I mean, great question.
There, exactly. There.
Yeah, yeah. Possibly, yeah, but I mean, like again, I mean, this needs to be like interpreted with caution, because scalp EEG source imaging has like a very washed out resolution. But yes, it was definitely most posterior than other sources. But yeah, it's a bit to be determined. But I mean, we were a bit surprised about this finding, but it was the most obvious one when comparing these two conditions. Yeah, so.
Okay, let's thank Martin.
Thank you.
And move on to our next and last speaker, Seong-Mi Son from University of California, San Francisco, on neural dynamics of dual task walking in Parkinson's disease. Yeah. Oh. Oops.
All right. Is it okay? Hello, everyone. My name is Song, and I'm so happy to share our research with you today. I'm a postdoc in one lab at UCSF. So how often do you find yourself walking without doing anything else? Oh, can you? I can see my slide here. So. All right, let's do it again. So how often do you find yourself walking without doing anything else? So probably very not often. So in our daily lives, so we often do something called dual tasking. So performing two tasks at the same time, such as checking your phone, chatting with your friends, or carrying grocery while you're walking. So for most of us, these kind of activities are very simple and routine activities that we hardly think about. But what happens when this seamless effortless start to break down, like people with the Parkinson's disease? So for individuals with the Parkinson's disease, it is not just a minor inconvenience. It becomes a daily, constant, and significant challenges that raise their risk of falling and consequently deeply affect their quality of life. So in Parkinson's disease, the brain communication network changes as the disease gets worse. So in healthy brain, the walking is automatic. So the brain motor control circuit system communicate smoothly with the minimal efforts. But in Parkinson's disease, the lack of dopamine that causes the faulty connection between the basal ganglia and motor cortex, and this creates an abnormal signal like the higher beta synchrony, which makes the decrease of the motor output. To make up for this, the brain reroutes the traffic. So it starts relying more on the cortical network rather than the subcortical to the cortical, and requiring the patient to use the cognitive input and the sensory input to able to walk. So gait and balance impairments are among the major cause of the mobility in Parkinson's disease. So these difficulties can appear in different ways. So trouble starting to walk, challenges in maintaining smooth rhythmic motion between both two legs, or difficulty adjusting step lengths or body position when something happens in environment. So as a result, up to 70% of Parkinson's disease patients experience the FARS, and making everyday mobility very risky and stressful. So what's more, when they try to walk while doing dual tasks, like our everyday activities. So during the dual task, individuals with the Parkinson's disease exhibit the noticeable decline in the walking performance. So walking speed and stride length significantly decrease, gait variability increase, meaning the step time and placement become less consistent. So in the brain side, the dual task situation are associated with the increase of the demand on the executive control system. So functional neuroimaging studies have shown the increased activation in the prefrontal cortex and the supplementary motor area during dual task condition compared to the single task condition. So this research suggests that individuals with the Parkinson's disease must recruit the more attention and the cognitive resources to maintain walking when they are multitasking. So thanks to the advanced deep brain stimulation technology, we can now directly record the neural signal from subcortical area, like the basal ganglia. But still, we do not fully understand how the brain network change during the dual task in the Parkinson's disease. And the previous research is not consistent. So our study aimed to investigate the neural circuit dynamics during dual task walking in individuals with Parkinson's disease to better understand the mechanism of the cognitive motor interference. So we hypothesized that alpha and beta band power will decrease during dual task walking, reflecting increase of the neural engagement. And our second hypothesis is the coherence between the brain network will increase during the dual task walking, indicating the stronger functional connectivity to support the added cognitive load. So we recruited the 4 patients with the PD, and each implanted the brain stimulation device with the Medtronic Summit RC+S. This is a research device that Doris already talked about today morning, and this device was capable of providing the therapeutic brain stimulation, simultaneously recording the neural signal with the Bluetooth communication. So during the data collection, the patient's device was set to their clinical settings, so providing the constant stimulation throughout the test. So we recorded the local field potential from the globus pallidus, which is part of the major ganglia, and ECOG, electrocorticography from the cortical surface overlying the primary motor cortex and premotor cortex. In addition to the neural recording, we captured the movement data using the IMU-based motion capture system, goniometer, and FSR sensor to evaluate the gait performance. So the patients completed the time up and go test, so commonly called as a TOG test. So this is widely used in clinical assessment to estimate the FAR risk. And the TOG test itself involved a sequence of everyday movement, including standing up, walking forward, turning around, and walking back, and finally sitting down. On the right side, the left side of the screen, you can see the motion capture video of the participant performing the TOG test. On the right side, it is the time frequency analysis showing the average data from the 4 subjects during the single TOG test. And in this plot, x-axis represent the time and y-axis represent the frequency. And this visualization illustrated how a power of the fluctuation occurs to different frequency band during the TOG test. So for this presentation, we'll specifically focus on the straight walking segment of the data. And to explore how adding cognitive impact walking, so we compare between the single and dual task condition. And during the dual task conditions, the patients did a counting backward by 3 from the given number. So let's look into the results. This is our gait metric results, and from left to the right side of the screen, the step length, walking speed, and step length symmetry index. So it is important to note that the symmetry index, the value close to the 0 is the more symmetrical. And the black bars are from the single task condition, and red bars from the dual task condition. And we observed a significant decrease in step length, walking speed, and the decrease of the step length symmetry. And this means the walking performance during the dual task was significantly decreased compared to the single task condition. So this is the neural results. And top of the screen here is the power spectrum density from a single subject. And x-axis here is the frequency, and y-axis the power. And each colored background is each frequency band, delta theta, alpha, beta, and gamma band. And red lines are from the result from the single task condition and dual task condition, and the black lines are from the single task condition. And at the bottom, we have a group level average during the dual task condition for each frequency band compared to the single task. So we observed the decrease of the alpha power from primary motor cortex and beta band power from the premotor cortex. And so as we may already know that when brain transition from the resting state to the active movement, alpha and beta power in the motor-related brain area typically decrease, reflecting the increase of the neural engagement. So in our study, like a dual task introduced additional cognitive load where they're like a walking, so increase their like a resource in allocation to support the simultaneous motor and cognitive processing. So this is a coherence result, and from top to bottom, we have a coherence between the globus pallidus and primary motor cortex, globus pallidus and premotor cortex, and primary motor cortex and premotor cortex. And x-axis is the frequency, and the y-axis the coherence value. And like the previous page, the black lines are from the single task condition, and red lines are from the dual task condition. And coherence analysis measures how strong the activity in the two brain regions is synchronized at each frequency. So this analysis help us to help us how different part of the brain communicate during the different tasks. So we observed the increase of the alpha and beta band coherence during the dual task condition compared to the single task condition. And we also observed this theta band activity between the globus pallidus and primary motor cortex during dual task condition compared to single task. And theta band activity is strongly associated with the higher level cognition, so such as the attention, error detection, or something like the executive control. So, and lastly, we did not observe any significant change in the coherence between the primary motor cortex and premotor cortex. So these results, our results suggest that the main point of the stronger connection during the dual task walking is in basal ganglia and motor cortex, but not within the motor cortex itself. So to sum up, so we found the dual task increase the cortical engagement. And also we found that dual task increase the cortical pallidal communication, but not within the motor cortex itself in the Parkinson's disease patients. And as a future research, we would like to explore other analysis metric, like a phase amplitude coupling or grandeur causality analysis. And we also want to expand our understanding with the wider range of brain network using the other modality like EEG or app-near system. And lastly, we want to extend our analysis beyond the walking to understand the everyday activities. And I'd like to thank our amazing patients and our amazing teams and funding resources. And thank you so much for your time today. Yeah, I'm happy to answer any question you may have. Thank you.
Any questions? There's no questions, so this concludes our session. Thanks again.
Okay.
There are no badges, but